I want to start just by acknowledging Ed, Denise, and Paul for organizing the conference. Again, another conference. I wasn't here last year. I was here a couple of years ago. And want to honor the work of a couple folks in the audience, perhaps even Dr. Downey. I may have some of your data. I didn't put your name there. That I'll show in my presentation who have more expertise than I do on some of this stuff. So, in the dose response society, the dose response world, things are very, very complex. We can take this to multiple levels of analysis. But for my talk today, I want to keep this really simple and kind of at a conceptual level, and then show you some supportive data here and there from different systems that address an underlying concept which I'm trying to get my head around, and would appreciate your feedback on it at the end. So, in the simplest sense, the stimulus response, the dose response, the challenge, and subsequent response, as Dr. Giordano mentioned, this is what we're faced with now. Independent of when you produce that challenge, pre or post conditioning, I guess is what I'm getting at here, versus when you actually see the response. Is it immediate? Is it delayed? I mean, really what we're talking about in terms of the challenge, the fundamental features is there's a magnitude to the stimulus and there's a duration to the stimulus. And this is pretty much how we kind of default to thinking about this whole thing. And together they produce a response, right? So we often think about this as an all or none thing. Did it work or did it not? And then we mess around with the magnitude and duration of the stimulus until we get something that works. So this is kind of a all or none thing. And that's kind of the mainstay of the field in many ways. So what I want to get at and talk about today a little bit is, how about the duration of the response? How can we affect this particular parameter? And really what I'm talking about, I guess, is the therapeutic window of this resilient state, of this disease-resistant state, this phenotype. And when you think about it that way, it's pretty obvious, but some of us are, including me, are very focused on just seeing, hey, we got an effect, we got a phenotype, and let's drill down, study the mechanisms, et cetera. So, what's really the problem, I guess, when we think about translation, is that if these phenotypes are short-lasting, how are we going to take these to patients? I mean, we can support ourselves, hopefully, with grant money and study this forever, but how are we going to get it into people? And even a long, long time ago, we knew this response was transient. Here's a paper from a couple decades ago. The fuzzy stippled bars here showing the days after a preconditioning dose of mild ischemia or brief ischemia, and the protection that you get against stroke. And you can see the protection kicks in at about 2 days, lasts up until 5 days. If you're waiting 5 days after the stimulus, but if you wait a week, you no longer see the protection. So we showed the same thing in the same time course here, up to a week after ischemia, ischemic preconditioning in this case. And you can see that you get fantastic protection. Here's in retinal ischemia, the B wave amplitude, a functional readout. You get fantastic protection if you wait, if you do the injurious ischemia 1 day or 3 days after the insult. But if you wait a week, you can see that there's absolutely this vertical hatch bar here, no protection at all. So it goes from basically fantastic, almost virtually full complete protection, 100% recovery, or protection against injury to nothing a week later. We did this in the mouse retina as well with brief ischemia, and in this case we even lose the protection within 3 days. So whether we measure the thickness of the different retinal layers or count the number of cells in different layers of the retina, it's kind of a complicated slide, but basically the bottom line is we lose the protection pretty quickly. So how can we extend the duration? And it seems like the way to think about this is the frequency of the challenge. And that is kind of what I want to spend the rest of the time talking about here. So in addition to showing you some New York Times bestseller books. So the very first step, the very first clue that maybe we could think about this and this would work is that you can re-trigger the conditioning response in the same tissue. And this was, again, another old paper here, but basically if you this is a global ischemia model looking at hippocampal CA1 injury, but if you precondition 30 days ahead of time, and then do your global ischemia, you don't get any protection. But if you do it again 3 days before, and you've done this 30 days previous in the same animal, you get full protection. In the retinal ischemia model I showed you earlier, again with the B wave amplitude here, same basic idea. If you precondition and wait 10 days, you don't get any protection shown down here. But if you do it again in a group of animals a day ahead of time, you can basically re-trigger the response and get a wonderful protection. So at least within a tissue, within a cell, what have you, we can reestablish the response, giving some hint perhaps that we can maintain a phenotype long term. So, sorry, that was the idea here is can we play with frequency and actually see if we can extend the duration of stroke tolerance or retinal ischemic tolerance, two models that I work in. And so here's the basic design. Normally, you precondition one time, you get a couple of days of protection, as I just reviewed for you. But what if we just kind of repetitively condition and then stop? Does this kind of therapeutic window here last a long time so that when you finally have your insult, you still can see a protective phenotype? And indeed, when we did this in a mouse stroke model, we were kind of surprised to find we preconditioned the animals with an intermittent repetitive hypoxic preconditioning protocol. Over a 2-week period, they had 9 bouts of hypoxia, relatively mild hypoxia, and basically took the animals back downstairs for a couple days all the way out to 2 months. And you can see here, this was kind of progressively how we did this. We brought them back up after a couple weeks. They were still protected, and ultimately 2 months later, still showing protection, doing absolutely nothing at all, doing the preconditioning for a couple weeks, and literally parking the animals in the vivarium for 2 months. And still they retain this phenotype. We see it in a retinal ischemia model as well. You can see it here structurally. Normally the retina thins, you lose all these cells in the ganglion cell layer and other layers, but we can see protection 4 weeks later. We can again quantify all this. I won't walk you through all this, but people in this field you can measure the thickness of the layers histologically, you can count cells, and basically you see that in the yellow bars here, fantastic protection a month later. You can do it functionally. Normally you lose the functional response in an electroretinogram, A wave and B wave response here with increasing light intensities. But if you precondition and wait a month, you get fantastic protection still if you repetitively precondition. I didn't find this paper until after we published our stuff, and if you caught the years on that, that was late 2000s, 2007, 2011. But this paper came out in 2004, and I didn't even see it until much after the fact. But looking at intermittent hypobaric preconditioning in rats, here's kind of their protocol. They did 7 weeks of intermittent hypobaric hypoxia, and then looked at infarct size in the heart. And you can see basically waiting a week, 35 days, or 90 days. At 35 days out after they stopped this, they still got protection against myocardial ischemia. Not at 90 days, but still at 35 days, 7 or 5 weeks out. So pretty impressive. So here's the paradigm then. We can repetitively precondition or challenge and get a long-lasting therapeutic window. So maybe instead of acute injury, something like I've been showing you here, stroke, heart attack, trauma, something where injury is fulminant and happens very quickly, can we take this into models of neurodegenerative disease where neurons are dying slowly, insipidly, chronically over a period of weeks to months in animal models, over a period of years in humans? And so that was the hypothesis, and emerging results suggest in some of these models here that this is actually working or can work. Here's what we did with glaucoma. This is a disease where the retinal ganglion cells in the front of the retina here, which have axons that form the optic nerve, these are the guys that die under high pressure in the eye, under high intraocular pressure. So in a mouse model, we induced intraocular pressure in one eye surgically by ligating episcleral veins, and then waited either 3 or 10 weeks after this preconditioning protocol here, and then looked at protection. And we looked at both the soma and the axons, because the axons are not, soma probably dies by apoptosis, axons are probably dying back or degenerating or disappearing by other unknown mechanisms. And it's interesting that the preconditioning insult, whether we did this 3-week cohort or this 10-week cohort, didn't change the intraocular pressure. So the retinal ganglion cells are still under the same injurious stress. The same injury magnitude is still there. So it's not why, but not why we'll get the protection that I'll show you in a second. And here's just looking at the soma protection, either at 3 weeks when we lost 21% of the cells, or out at 10 weeks where we've now lost 30, kind of a progressive loss of retinal ganglion cell soma. Fantastic protection if we repetitively precondition ahead of time because of that long therapeutic window. If you look out in the nerve, you can do PPD immunostaining, and basically here's the messed up glaucoma optic nerve and the preconditioned one as an example. But same idea, pretty significant loss of axons in the optic nerve, but wonderful protection if you repetitively precondition. And even in the axons that are in the what they call the nerve fiber layer, these are the retinal ganglion cell axons proximal, close to the cell body, that are still in the retina before they form the optic nerve. You can quantify these guys with immunofluorescence, and you can see it appears that there's loss that's pretty robust out here of about half of them sort of falling away with this SMI32 immunostain, at least a marker of neurofilaments. But again, wonderful protection. So we did it. We can show protection against a chronic neurodegenerative disease, but it's preconditioning. So how are we going to, clinically, it's conceptually cool, but not that translationally wonderful. So bench to bedside, maybe what we need to do is I don't know if this is called per-conditioning or post-conditioning, but treat during disease. Not get too hung up on the semantics, but the idea of we can induce and sustain a protective effect, a protective phenotype during disease progression. And so we took that same episcleral vein ligation model of a glaucoma in the mouse. We induced this increase in pressure and then waited a couple of days before we started the hypoxic challenge, and shown here are the times we did that over a 3-week period. And again, didn't change the pressure at all, so the cells are still facing the same insult. But now looking again at the protection of the soma, we see some loss there that's wonderfully protected against. Looking out in the nerve, same idea, great protection. Not quite as robust as the preconditioning, but still significant. We can do this functionally with visual evoked potential amplitudes and looking at visual acuity, and you can see basically the qualitative point I'm trying to drive home to you here is we can still get protection even during disease. We can kind of either slow down the kinetics of the disease or rescue. Not quite sure what the semantics, the most appropriate semantics would be, but conceptually it's working. Another group did this with diabetic retinopathy, where they intermittently challenged rats during the disease, and you can see that normally the electrophysiologic response of the retina fades over time. I don't know if you can see that in the back, but this is a 10-week time course here. The A wave and B wave of the electroretinogram fading away as the retinopathy progresses, but when they preconditioned the cohorts, they got good protection, functional protection. And even looking out here, starting at 6 weeks with the idea of rescue, here's the data they got there, same thing. So they kind of halted the disease course midstream, and were able to basically stall out the disease with an intermittent approach like this. We did this isn't published, but we did this in a mouse model of multiple sclerosis where you immunize against myelin and you can score the animals a few days after the immunization. They get pretty sick, and you do a neurologic deficit, kind of neurologic injury score, behavior score. And if we per-condition those guys with intermittent hypoxia, we delay the onset. They ultimately reach the same severity, and this is a model that's not relapsing, remitting like human MS. There are some other mouse models that are. We haven't tried that yet. But basically delay the onset. So it's a kinetic change that we had success with here that in humans, it's only a few days here, nine days or so, but maybe this could translate to an overall big difference in the human. And we also, looking in the spinal cord, you can see less inflammation. You start looking at phenotypes, what's going on, what's accounting for the protection. In a mouse model of ALS, there's a transgenic mouse, this SOD G93A transgenic, that basically is a kind of a bread and butter model for ALS in that world. And the animals, basically you just monitor the body weight. It's quite simple, but they grow normally and they keep gaining weight, and at a certain point they start losing weight just like humans and start the muscle atrophies, et cetera. And we found that if we repetitively precondition or per-condition or post-condition during the disease in these guys, we get delays in the onset of disease and delay in time of death. So something's going on. We're still affecting disease kinetics here. This is a paper that Dr. Ding was co-author on. He's going to speak later, where they used a remote conditioning, and apologies to Dr. Reddington too, who has a different device for doing this. This is a study out of China, but basically a bilateral blood pressure cuff approach to do intermittent inflation, deflation, inflation, deflation. They've done this in animals, 5 minutes on, 5 minutes off, kind of taken these protocols and moved them into people because it appears to be harmless. Here's just a schematic showing a leg cuff. The idea of maybe we need to, the protection may be dependent on how much skeletal muscle mass is rendered ischemic, hence the bilateral approach here. But anyway, it's thought to be either humourally mated, something released into the bloodstream, a mix of cytokines perhaps, we have no idea, and there may be a neural component as well. And they did this in patients that already had a stroke. They had symptomatic intracarotid atherosclerotic disease. And when they did this for 300 consecutive days, twice a day in these patients, they probably, maybe Dr. Ding can speak whether they got paid to do this, but I would imagine the compliance was pretty good. They got great reduction in recurrence of stroke, the number of TIAs these patients experienced, and stroke recovery was improved significantly, so they returned to kind of baseline functional scores much quicker in the group that got this treatment. So it's, again, something about this repetitive treatment. This is existing disease, post-stroke, maybe preconditioning for a second stroke, recurrent stroke. Again, the semantics can confuse sometimes more than they can help, but very powerful outcome. So I was already kind of alluding to this idea. We can probably use this repetitive conditioning, whether it's hypoxia or not, as a recovery therapy. Dr. Giordano kind of alluded to that as well, or a rescue therapy. I've shown some data. And there are other diseases and models out there. I'll just show you a couple examples about. They're quite interesting, quite robust effects. This is by Dr. Gordon Mitchell's group that he's going to speak at this meeting. But they basically had a spinal cord injury model in rats, and they did hypoxic post-conditioning 1 time a day for 7 consecutive days after the injury. And this is the correct steps on this kind of ladder thing as the rat moves across it. And here's their treatment protocol here for 7 days in a row, just 1 time a day. But you can see the big increase they got in proper or correct placement of the foot on the ladder. And one of the more powerful things, the reason I'm kind of showing this slide in part is because even 3 weeks later, they still sustained the effect after the treatment was over. So again, there's something about maybe repetitive conditioning that allows the phenotype to persist and hold, so to speak. There was a group looking at focal stroke in mice, and they did this starting 5 days after stroke, mild hypoxic challenges. Here's the protocol they used for 6 weeks. And they discovered that there was less thalamic atrophy in these animals way out at the 6-week time point. So there's a kind of an accumulative effect. And people are looking too at not only just kind of histology and function and lesion size and morphology of the infarction, but also kind of functional outcomes from stroke models, global ischemia, et cetera, and using Morris water maze. I don't know if I'm familiar with some of this. Some in the audience probably are, but you can basically significantly affect the amount of time that the animal takes to find this platform with intermittent hypoxic challenges. Here's the protocol they used here, and as well as the time spent in the quadrant where the platform was submerged and they couldn't see it, but had previously trained to know where that was based on cues. So you can see that a significant increase in the time where they know where to go if they got this treatment post ischemia. And of course, they got wonderful histology to go along with that, showing that they protected the CA1 hippocampal neurons in the intermittent hypoxia protocol. These are the ones that are most sensitive to a global ischemic cardiac arrest type insult. This is, even intermittent hypoxia has been shown in an existing hypertension to reduce blood pressure, both systolic and diastolic. These are patients, human now, that had disease either less than 5 years duration or greater than 5 years duration. Two groups are shown here. And you can see compared to the controls here in the dark bars, you can see how the blood pressure was lowered with an intermittent hypoxic protocol. These are patients that are not on any existing medications, but even if they were hypertensive for a long time, greater than 5 years, they still got an effect of this intermittent hypoxic challenge. And again, interestingly, the effect persisted in 85% of these treated individuals when measured 3 months later. So these data were right at the end of the treatment, at the end of 20 days, but 3 months later. So they're holding on to the phenotype again. This is a paper by Dr. Reddington's group in an MI model in the mouse. And here's some of the protocols they used, per conditioning right during the middle of the ischemia. I was just going to point out these two protocols where they post-conditioned every 3 days, basically, or every day, and compared the two, and really got it's a beautiful paper. They got a nice dose response here, looking at the Kaplan-Meier curve. This is the everyday group, and this is the every third day group. This is the controls down here. This is just the anesthetic control. And you can even see when they looked at macrophage numbers and neutrophils here in the two bars highlighted above the red bar here, they got, again, a dose-dependent response, whether the post-conditioning was daily or every 3 days. No change in infarct, but other stuff going on. I mean, still protected, both of them protected, but clearly something going on here with other phenotypic measures. So I'm just kind of going to leave this here. In contrast to this kind of very pattern, they're very predictable, consistent pattern, right, of every day or every 3 days. Maybe and nobody's done this, we haven't tried this, but maybe we can do a volley of challenges and then the phenotype changes and is held onto, so to speak, persists and gradually fades, and then we can maybe do this again, reintroduce that phenotype and hold on to it for a while. So maybe it doesn't have to be literally constant. This, from what I can find in the literature, I've not been able to see any studies that have tried to look at this systematically. Okay, a longer, even still, kind of phenotypic response is a result of, there's a lot of this in literature, actually conditioning neonates or the mothers when babies are in the uterine development or even during early development, and seeing beneficial phenotypes in adulthood. And there's a lot of data on this with some very interesting results, but I just didn't want to throw a bunch of graphs at you. So just a couple of examples here where exercising the pregnant mother, for example, promotes resilience to stress in the babies when they're grown up. So, we're talking months and months of phenotypic resilience here. Intermittent hypoxic challenges during the first month of life in rats and mice improve learning and memory, all kinds of other related effects that are beneficial. And you can even, in monkeys, it's maternal separation, stress, new pairings, that kind of thing. Stress that, again, is a challenge that's, but maybe not harmful in and of itself, has long-term effects that can be witnessed or measured at the adult stage. So, and these are relatively transient challenges, 1 month or something like that, and then seeing it 6-7 months later without any intervention. So kind of similar to what I had showed you earlier with our results with stroke and in the retinal models, that Mother Nature is basically holding on to this phenotype. And I guess the longest, perhaps the longest example of that would be whether you can pass on an adaptive response to your children. And kind of science fiction-like, I guess, but this is actually a transgenerational model, and we tested it in our in retinal ischemia again. We did 4 months of repetitive hypoxic preconditioning in males and females. They were separate. Then we put them together to breed. They got pregnant. They had births. We let the F1 generation grow up, and then we did retinal ischemia and measured electroretinography as a functional readout. We had done this for so many years, so for us, this was a very reproducible, standard, dependable way to cause injury and to see protection if it was there. And indeed, looking at the waveforms here, again, the A wave and B wave, the electroretinogram, normally you'd lose about 50% of that response, what I showed you earlier, but you can see in these babies whose parents got preconditioned, they're showing significant protective, functional protection. So mechanisms. Dr. Giordano talked about this quite a bit. Whether, I'm sort of the feeling that maybe we may learn about new molecular targets or drug targets by understanding mechanisms. On the other hand, you might argue that with those blood pressure cuff approaches or hypoxia or hyperoxia, it would appear there's no harm, or we just have to make sure there's no harm in the stimulus. Then maybe we can start getting this into the clinic and not worry about how it happens, because we'll probably spend decades trying to parse mechanisms. It's a personal editorial there, but anyway, but you can look at phenotypes as one kind of reverse backwards approach. What does the protected animal, in this case, or human, look like? Ultimately, and the front end of things, it's probably a bunch of signal transduction stuff that activates transcription factors, and then overall, this is an epigenetic response. It's classic. All this pre and post conditioning in hormesis is really epigenetics, in my view. And so let me just show you. I showed you this thalamic atrophy reduction in this repetitive conditioning model in rats before, or sorry, in mice before. But this same group did this. This was post conditioning now after the insult focal stroke, but they did this same protocol just to understand what happens without any stroke at all. And here's the time points: 1 day, 25 days, and 41 days, after this kind of protocol of intermittent hypoxia. And you can see, just measuring adrenomedullin levels in the brain, they got increases that were sort of time dependent. So things are changing over time as you repetitively condition. It's not a static response. Look at VEGF here. Nothing really changed at all until you're way out here, 6-7 weeks in, and now VEGF starts going up. Even though you've been doing the same thing for 5 weeks, nothing happens. So there's thresholds, there's things like that happening that we have probably no clue about, really. Here's another paper from Dr. Reddington's group where they did remote conditioning with the blood pressure cuffs in humans, took the neutrophils and looked at some of their phenotypes, either 1 day after a single cuff inflation or after 10 days of cuff inflation, daily cuff inflation. And you can see, for example, maybe this green bar here, but basically measuring cytokine production by the neutrophils. Nothing immediately, but after 10 cycles, a big increase in IL-10 production. The phagocytic activity of the neutrophils changes immediately. There's maybe a little bit of an increase, but 10 days later is a reduction. So these phenotypes are changing over time. I won't get into this too much, but for me, I think a lot of this has to do at a metabolic level with downregulation of a number of metabolic genes, protein synthesis genes, et cetera. It's been looked at in hypoxia-tolerant fruit flies, that there's a huge downregulation in particularly the oxphos stuff, and this has all been studied in detail so that basically the animal downregulates its metabolism. It's been done in worms. We'll hear more about that, I think, right after my talk. And there was a mutant discovered that showed a wonderful hypoxic tolerance, and it turns out this mutant worm was missing a tRNA synthetase. So basically they were, like, it was a translational arrest kind of phenotype. They weren't making proteins, and they could no longer need, have to use up so much ATP to do that. This is the same kind of translational arrest that happens with hibernation, that happens in turtles and carp and things like that that can survive anoxia. And if you look at high altitude Quechuan Indians in the Andes, for example, and do PET scans on these guys, normally here's a normal sea level control. And when you look at one of these individuals who lives at 16, 18,000 feet, they're extremely metabolically downregulated, these brain scans here. This looks practically comatose compared to normal. And here's the interesting thing: when they brought those people, the same individuals, down to sea level, for them it was like a relative hyperoxia. They had more oxygen than they knew what to do with. They were used to very low oxygen, so they downregulated even more. So there's something going on. This may have some parallels too with hypothermia protection. Okay, so anyway, a little aside there, but transcription factors that are driving the epigenetic response, there's a bunch of them for hypoxia. Of course, hypoxia is not the only thing that will trigger these responses. But when we looked at HIF, for example, and it's interesting if we compare a single hypoxic challenge versus a repetitive one. A single one, here's a time scale here out in days. A single challenge looking at retinal levels of HIF elevates HIF right away. But after 6 of these things, you get a much bigger increase in HIF, and it lasts a little bit longer. And if you look at a HIF gene product like hemoxygenase 1, in both cases you get basically the same magnitude of increase, but look what happens with the repetitive preconditioning here is that you still, out to a month later, you still have high levels of HO1 1 month after this little volley of preconditioning. So something's going, this HIF drives a couple hundred different genes, not just HO1. So, but we're getting both, transcriptional changes as well as target gene changes. Delta FOSB is another possibility it's really more popular in the addiction literature, but it looks like this particular transcription factor accumulates after a single challenge. You can see low level accumulating here of these certain isoforms. This combines with June and becomes part of the AP1 complex. It's either a transcriptional activator or repressor. And if you do repetitive stimulations, you can see it basically just accumulates and lasts well after the stimulus is gone. This is work by Eric Nestler at Mount Sinai. So there are probably other examples of this. We just haven't figured out what they are, where the response, the transcription factor regulation is affected by the intermittent stress. So let me close with, again, what I hinted at before. Really, this is, I think, all epigenetics, the idea that you change gene expression, you modify the DNA, so you can either get immediate or delayed changes in gene expression and thus phenotype. The 3 main ways that we understand right now that this happens is DNA is methylated, or there's post-translational modifications of the histones that allow exposure to the genes, and not shown here, I don't think, are some RNI-mediated regulation, long non-coding RNIs that also orchestrate some of this. So maybe not to introduce another term into the lexicon, but maybe adaptive epigenetics is really kind of what this whole thing is about, what this whole meeting is about. It's the idea that we're changing gene expression and getting changes in phenotype, potentially long-lasting, that can affect resilience or health. So let me stop there, acknowledge the funding sources, and happy to take questions. So we're in the middle of analyzing, we collected sperm from the mice that were treated in the transgenerational study for 4 months and did a genome-wide DNA methylome on them. It's a big data problem now because we got Excel spreadsheets with 20,000 rows of data on them. So we're in the middle of the bioinformatic analysis on that, but it looks like transposon, there's a transposome. The transposons are changing, so and other data that we're still trying to pull out of that. But and we can't do that on the germ cells of the mother. It's much harder to collect eggs, get enough eggs to do. But we haven't done it in other ways, no. Because there's epigenetic changes in the target tissue, so to speak, in the brain, retina, heart, wherever you're looking.
Have you seen, so you don't know, you don't have any results yet?
Not yet, no.
And then next, just as a follow-up, is what happens if you exercise? Would exercise do the decline of epigenetic changes? Would exercising the rats do the same, create the same kinds of changes? Have you thought about doing that? Or does exercising rats give you the same protection against your endpoints that you manifest in?
Yeah, those studies have been done. The question is, if you could all hear that, is exercise as a conditioning stimulus, does it do kind of some of the things we're talking about here? And indeed, those studies, we haven't done that in our lab, but those studies have been published. It's well known to reduce stroke volume in animal models and rodent models.
Do they have epigenetic data with the exercising?
I don't think so, no.
So they haven't done the epigenetic
No. And there was a paper that got a lot of press in the New York Times and the lay press and stuff about exercise in protecting photoreceptors against light damage and in models of retinitis pigmentosa and that kind of thing, maybe protecting against blindness, just simply exercising the animals.
Very, very nice thought. One of the things, trying to link exercise and the hypoxia you talked about, they're very different in one way. The brain goes hypoxic during systemic hypoxia. It does not during exercise. What is common is they both activate monoaminergic systems. And that links in with vagal stimulation and all kinds of other deep brain stimulations. Do you think that's more what we're talking about here when you're talking about at least these neural adaptive responses?
Conceivably, sure. It's all in the realm of possibility.
But not much.
Yeah.
The other part of it is just the histone modifications are pretty likely in response to hypoxia per se.
Sure.
Have you looked at that at all?
And we haven't.
Have you really done that?
We haven't done that yet either. We are starting a collaboration to do that with some epigenetic folks, but we haven't done it yet. Thank you. Yeah.
You're primarily using hypoxia and remote preconditioning, neither of which we're really sure exactly what the mechanisms are. Can you do that with ischemic preconditioning, with like an includer on a coronary or something like that, where a pathway that we know a little bit more about?
Do repetitive, basically?
So like a biopsy every day.
Yeah.
Includer. Has that been done?
I don't think that's been done.
How about hyperbaric? Is it hyperbaric?
We'll let George address that.
Repetitive, instead of a hypoxic every day, a hyperbaric.
Yeah, this is all the talking dose, right? I'm interested to know how we dose hypoxia in line 11%. But, when you feel like hyperbarics, they see a robust effect in terms of conditioning. But it all these different algorithms in terms of molecular slides, staccato. You do a couple, wait a while, and do a couple. It's not going to work out. What is the timing and dose of the stimuli?
We typically use males, but the literature is suggesting there may be some, now people that are starting to look at this, of course, and there's age is another factor. All the comorbidities, of course, play into this shifting the dose response, but yeah. I'm not sure if you could hear that, but a long question about male-female differences, basically sex hormones driving phenotypes and what have you. We have not done that much at all, no. And the only data I have related to this repetitive idea is in that ALS model. We parsed the data by sex, and the females did not have as robust of a response. And that's pilot data. We haven't published that. So, but the trend was pretty clear. Whatever that means, I don't know. No.
Do these retinal preconditioning effects, do they require a systemic response? That is, if you were to culture the retina, can the retina itself mount this response, or does it require a systemic response?
Well, I think in all these models, you can culture cells, and cells have the innate ability to become conditioned and change their phenotype.
Do you expect this is a direct effect on the retinal ganglion cells?
Oh, well, so it's hard to say. It's hard to say. So the question is, in an example with retinal ganglion cell protection, say in glaucoma, is it a specific effect on that cell? No idea, really. You know how EPO comes from the kidney? We're systemically exposing these animals to hypoxia, whole body hyperoxia, exercise. A lot of these stimuli are whole body, at the whole body level, right? So there may be certain tissues that are releasing the right kinds of cytokines, et cetera, that are accounting for adaptation or adaptive epigenetic responses in all the body's different tissues. That's what the blood pressure cuff data is suggesting. And of course, it invokes multiple layers of signaling, paracrine signaling. Are the endothelial cells the one that's the sentry cell that's understanding the mix of cytokines in the blood that goes by and interpreting that as a danger signal or as an adaptive signal, and then in turn kind of turning around and telling the parenchymal cell, the myocyte, the neuron, what have you, hey, you need to adapt because I just saw this danger signal. So there's intercellular communication as part of this. I think that's part of the fun of this whole thing is that it's systemic, but you can tease out, of course, mechanisms in a cell culture dish. Yes?
Very interesting presentation. I love it. A couple comments. One is that you mentioned transposons, and I would suggest that part of the stress response to the upgrading of transposable elements, it absolutely always happens, and its effect on the genome can be very, very profound. We don't understand much about that, but I think it is really an important forefront area to mention along with your histones and methylation stuff. The other thing was a question you caught my attention when you mentioned the importance of repeating stresses in the presence of an injury or the presence of a condition. And I wonder whether our whole attitude towards treatment of infectious diseases could be affected by this, whether we could adopt stresses for that. I mean, can you speculate on that?
So the question was, can a repetitive kind of therapeutic approach that I talked about here with ongoing disease be applicable to infectious disease kind of paradigms? Sure, I guess. You know, it's almost like a vaccine-like kind of thing, right? It's some of the data I show with treating the younger during development and then kind of having immunity to injury or more resilient kind of phenotypes later in life. So sure.
So we have to start telling people, stop taking it easy.
Yeah.
Right.
Yes. Okay. I think we might have to go on.
What-
Oh,
quick
Dr. Biggs,
question.
one more.
Did you use hypoxia model in your lab?
Yes.
Or ischemia model for conditioning?
We played a little bit with ischemia a long time ago, but now the bread and butter approach is this intermittent hypoxia. Yes.
So what do you do? Use this model?
How do we do it?
Yeah, how do you do it?
So we actually put the animals into a hypoxic chamber where we can control the gas tension for,
How
and
long?
so the whole animal is being exposed.